It's my pleasure to
You're
present to
welcome.
you a part of the work I've done throughout my thesis, and in particular, I would like to introduce you to the method of persistent homology. Um, yeah, um, which is persistent homology, which is a branch of topological data analysis. And what is topological data analysis? So basically, the problematic of we are facing today is the amount of data we are facing in our everyday life. And this data could be represented, for example, by a scanned 3D object. So we can take an object, scan it, and have the coordinates XYZ in the space. This observation or another observation would be like data as form of galaxies. So the position of each galaxy in the space um, could be also studied, or the data could be an image or like a face, and we will try to recognize the facial expression of an individual to analyze whether he's happy, he's sad, so on. Another form of data could be like a vector. For example, in your smartphone, while walking, you can have the speed of your walking, and by analyzing, for example, taking this observation throughout the day, we can analyze what type of activity you did, whether you run, you walked, you did sports, you climbed. So yeah, we are like around us and everyday life, there are data that could be represented often as a metric space, and the idea is to analyze this data, the shape of this data, to extract relevant topological features and take advantage of this information for later on for tasks like classification, recognition, machine learning, or prediction, for prediction, uh, whatever prediction we want to do. So I will give like some sort of example from real life. This is the first study that used topological data analysis where to medicine. Here we have each patient is represented by a genome, a vector of dimension 262, and by analyzing a group of patients that have breast cancer, we will obtain a point cloud in R to the power of 262. And by analyzing this point cloud, which is a huge point cloud in a high dimensional space, they analyzed the shape of this point cloud and discovered a new type of breast cancer, and further analysis were done on this point cloud to um, to discover this type of breast cancer, and earlier recognition of this type of cancer could lead to a survival of 100%. Another example is in chemistry. For example, this is a molecule, cyclooctane, that contains 24 atoms, and each atom is represented by a three-dimensional coordinate. So we will have a point cloud in R to the power of 72. And by taking a collection of such molecules, the authors have to analyze what shape does these molecules take, because they can have different shapes. And by analyzing a lot of molecules, they discovered that they have the shape, the union of a sphere with a plain bottle. So I'm not going into the details of this example, but just to give you an introduction of the idea that what we are interested in this type of questions. What is the shape of data? And this is the purpose of topological data analysis, to discover the shape of data and what topological features we can extract from this data. Like here, we discovered that, or the author discovered that the data has a shape of a sphere plus a clean bottle. And this is the main object of topological data analysis, to know what is the shape of data. Another example from real life, which is this painting, pointillism, we call it. So the painting is um, um, has the shape of dots. We are not the artist doesn't draw a continuous shape, but he draws dots. And just like our brain can visualize the shapes in this painting, like the little boat, the tree, the kayak. So this is the idea of topological data analysis. From the point cloud, we can reconstruct the shape that the point cloud represents. And to do this, we have to do some algebraic topology. So basically, topological data analysis is a branch of algebraic topology that is used to study topological space. And the goal is to find algebraic invariants to classify topological spaces up to homeomorphisms. So, homeomorphism between two topological space is a continuous function whose inverse exists and is also continuous. Like one famous example of two homeomorphic spaces are the mug, the coffee mug, and the donut. So basically, this joke that everyone does, that topologists do not know the difference between a mug and a donut. So basically, a homeomorphism means that we can stretch, bend, deform any space, but we cannot like cut the space or glue the space. And by this homeomorphism, we can know that we can deform the mug in order to obtain a donut. So another example of two homeomorphic spaces would be the circles, the space of circles and the space of squares. Throughout this application, we can project each point radially to the square, and the inverse is also correct. But a circle is not homeomorphic to an interval, because in order from a circle to have an interval, we have to cut it and make it an interval, which is not allowed to cut. It's not allowed to cut. It is surely it is a lot harder to prove this fact, but the idea is that in order from a circle to obtain an interval, we have to cut it, which is not allowed to cut spaces. And using this homeomorphism, we can classify shapes into homeomorphism classes. So we have the class of circles, we have the class of intervals, we have the class of crosses, we have the class of spheres. So those are in the same class, the shape in the same class, or the spaces in the same class are said to be homeomorphic. Now, in this case, we can like easily see that these shapes are homeomorphic. Like a square could be deformed into a circle could be deformed into a triangle. In the case of intervals, an interval could be like rotated or translated. It could be deformed. It is easy to see that those spaces are homeomorphic. But if we take like these two shapes, it is not very easy to say that if these spaces are homeomorphic. And this is why we have to use the notion of invariants. So an algebraic invariant is a property that remains unchanged by any transformation. For example, the sum of the angles of a triangle, like you can rotate it, deform the triangle, the sum of the angles would remain 180. The dimension of a vector space, the determinant of a matrix, a characteristic that is called Euler-Poincaré, and what we call Betti numbers, which we are interested in. So basically, Betti numbers are, let's say informally, are the number of k-dimensional holes in a topological space. For example, beta 0 would be the number of connected components in a space, beta 1 would be the number of cycles, and beta 2 would be the number of cavities. Like a point, like this one, we know that a point is one connected component. It does not contain any hole, it does not contain a cavity, it does not contain a cycle. The circle is one connected component. It contains a hole, so it is like if you want to reduce a circle, you cannot reduce it to a point. So it contains a hole of dimension 1. It contains a cycle, but it does not contain a cavity. A sphere also contains one connected component. It doesn't contain any cycles because if you want to draw a cycle on the sphere, you can at any time reduce it to a point. However, if you want to draw a cycle inside the sphere, you cannot reduce it to a point. So this means that the sphere contains a cavity. And the torus, which is a little bit more complicated to see, it contains one connected component. It is one connected component. It contains two cycles because if you take this cycle in red here, you cannot reduce it to a point. And if you take like the vertical cycle in red, you cannot reduce it to a point. And the horizontal cycle, which is in purple here or in pink, also you cannot reduce it to a cycle, to a point, sorry. So a torus contains two cycles, and since there is a hole inside it, a void, so it contains a cavity, so beta 2 is equal to 1. And we are interested in those Betti numbers because to decide whether two spaces are homeomorphic, if they do not have the same Betti number in each dimension, it means they are not homeomorphic. So this is an algebraic invariant. And basically, how can we compute it without going into the details? We need to construct a simplicial complex, so which is a combinatorial description of the space. Using linear algebra, we can compute the homology group of the space, and the dimension of the homology group would be equal to the Betti numbers. And for example, so since we are interested in point clouds, as I said, we are interested in data which are represented by point clouds. We are interested in the Betti numbers of point clouds and how to access the topological characteristics in point clouds. So if we see this point cloud, it contains 20 points, so it contains 20 connected components. But we do not see any further dimension. We do not see cycles, we do not see cavities as this point clouds as it is. But our brain can see that this point cloud has the shape of two cycles, one big cycle and another little cycle glued to it. So we can have the intuition that this point cloud represents one connected component and two cycles. And we want to formalize this idea. And to do this, we need persistent homology. We need persistent homology to describe the topological features of this point clouds beyond just the 20 connected components. So persistent homology, as I said, is a branch of topological data analysis, and it appeared in the late 90s, and it's booming right now in terms of application to biological data and other area of sciences. And the goal of persistent homology is to determine, describe, and compare the topological characteristics of data. What we mean by topological characteristics is exactly the connected components, cycles, cavities, which are in dimensions 0, 1, and 2, and so on in other dimensions. And to describe those topological features, we want to use topological descriptors. One topological descriptor that is very common in persistent homology, which I will introduce you in a bit, is what we call barcodes. And to compare point clouds, instead of comparing them directly, like we can take 2 point clouds and like, I don't know, compute an Euclidean distance, but we don't want to do this because we don't like want the metric that represent the point cloud, but we want to compare the topological characteristics of these point clouds. That is why we have distances that can compare the barcodes. For instance, the bottleneck distance and the Wasserstein distance, also that I will explain to you how to what those distances are. So the general pipeline of persistent homology is true. Like I am interested to study a point cloud. To do this, I don't have access to topological features, so I want to apply a filtration process to generate a combinatorial object that presents this point cloud and that give me access to higher dimensional topological characteristics other than connected components. And I want to visualize those topological characteristics in a representation that we call barcode. So I will explain to you how to, from a point cloud, obtain this geometric representation, which is the barcode. So basically, we want to represent the point cloud by a simplicial complex. So for those who don't know, a simplicial complex is a geometrical object or a combinatorial object that is formed from simplices. A zero simplex is the point, a vertex. A one simplex is an edge. A two simplex is a triangle. A three simplex is a tetrahedron, and so on in other dimensions. And the simplicial complex is formed by those simplices by gluing simplices under certain conditions. So we have to glue those simplices on a common face. For example, here we are gluing this triangle and this triangle on a common one simplex. I cannot glue this triangle over this triangle. It doesn't make any sense. We have to have an intersection, which is a face of the two, or the intersection can be empty. For example, like this triangle and this point, the intersection between them is an empty set. So basically, this is a simplicial complex. But the question is how to obtain a simplicial complex from a point cloud. The idea is to take a ball centered at each point and make the radius of these balls grow. Each time we have the intersection between two balls, we have to add the one simplex that join the centers. Like here, we have the intersection between these two balls. We add this edge between the centers. Here also we have the intersection. We add the edge between them. If we have the intersection between two balls, we add the triangle. For example, here we have the intersection between three balls. We add the triangle like here. If we have an intersection between four balls, we add a tetrahedron, and so on in other dimensions. By making the radius grow, at each time we have intersection, we have to add a simplex. And now the question is, okay, I have this point cloud. I want to represent it by a simplicial complex. But what simplicial complex should I take? What radius should I take? Because I said we can take a ball and a ball with a radius, and according to this radius, I will add the simplices. And the question is, what radius should be like the considered one? And this is the particularity of persistent homology, that it does not precise a single radius, but the idea is to look at all possible radius and see how the topological features will vary from radius 0 to the maximal radius. So this is the idea of applying a filtration process. We begin by the point cloud, which is at step 0. At step 0, we have 20 points, so 20 connected components, and represented by 20 interval in the barcode. And as I said, we want to take the ball centered at each point and make the radius grow. As the radius grow, if we have the intersection between two balls, we will add the one simplex that join the centers. So when we do this, we have one connected component that dies. And when it dies, so we stop the interval and we have 19 other connected components that persist and which are represented by the persistence of the 19 other intervals. We go, we increase the radius, the points begin to connect, and as points connect, we have connected components that dies. So we see like intervals stopped throughout the filtration process. At this point, at this step of the filtration, we have 10 connected components, which are represented by 10 intervals in the barcode, to 10 persistent intervals at this stage. We increase a little bit more the radius. At this step of the filtration, we have 4 connected components that are represented by 4 intervals in the barcode, 4 persistent interval in the barcodes. And what we observe more is the appearance of a little cycle here that is represented by this bar in the barcode. As we increase a little bit more, at this step of the filtration, all points are connected. So we have one connected component that is represented by this interval that will persist all along. The cycle that appeared in the filtration before here disappears, so we stop the interval and we see that another cycle appear, a bigger cycle appear that is represented by this little interval on the barcode. When the radius is like big enough or large enough, at the end of the filtration, so this is the last step of the filtration, the cycle that was here disappears, and this is represented by like we stop the interval when the cycle disappears, and we obtain the barcode that represent the this point cloud. So basically, what the barcode encodes the evolution of topological characteristic throughout the filtration, the appearance and disappearance of topological characteristics. And remember what I said earlier. In this point cloud, we have the intuition that there is two cycles, one big cycle, one little cycle, and we have two connected components. And this barcode confirm what we saw. So we have this large interval here represent the big cycle, and this little interval right here represents the little cycle and the point cloud. And we have this interval, the large interval represent that we have one connected component one connected component. So this is a simple example, which is the barcode is like very easy, and the point cloud is only in R2. We can imagine for a higher dimension point cloud, we will see other topological characteristics in dimension 2, like cavities and so on. So with this process, we can obtain a geometric representation of the point cloud. And algebraically, this is equivalent. So the filtration process is a sequence of simplicial complex, a nested sequence of simplicial complex to which we apply the homology group in dimension k. In dimension k, and this generates what we call the persistent model, which by the decomposition theorem is decomposed to what we call a p-interval that gives us the intervals in the barcode. So yeah, basically this is the persistent homology pipeline. And one particularity of persistent homology is that it persistent homology can capture the real signal in the point cloud. So here, if we have a point cloud that is a little bit noisy, we can still, with the noise will be represented by short intervals, and the large intervals will represent real signal. Like here, we have little intervals, like very short intervals, that represent the fact that this point cloud is not a perfect circle, but a noisy circle. And why do we know that it is a circle? Because we have a large interval in dimension 1 that represent the cycle in the point cloud. And these short intervals represent the fact that this point cloud is noisy. And one thing that I didn't mention is that persistent homology verifies a stability theorem, saying that um, even though we have noise in the point cloud, we know that the topological features that we capture in the barcode are stable under noise. Yeah, we can talk about this theorem later on, but the idea is that we know that even though there is noise in the point cloud, we can know that what we are capturing in the barcode is the real signal of the point cloud. And this is very important in applications like biology because we cannot be certain of the quality of the objects we are analyzing, especially if they are experimental. They could be like biases in the point clouds we are analyzing. Now, as I said, the idea is also to be able to compare point clouds. And to compare point clouds, we have we want to compare their topological features, so compare their barcodes. And one way to do is the bottleneck distance. So the goal is to compare two barcodes by computing the distance between them. And to do this, we have to define a matching between the intervals of two barcodes. And this matching should be defined in an optimal way. It means that here we have two intervals in this barcode, two intervals in the other barcode, and we have to find an optimal matching. It means that we have to match intervals that are matched. It means that this A1, I will match it with this one because the length of it is similar. I will not match it with this short interval because the idea is to find the minimal cost of this matching. And once we match the interval, we will take the difference between the matched interval, which is the difference between the extremities of the intervals, and we will take the maximum between the differences. So here we have the maximum between x, y, z because 0 and z. 0 because the two matched intervals both begin at 6. And we take the maximal value between them. So basically, this is what the formula will mean. And when we do this, we take the maximum between those four values, and then we take the infimum over all possible matching. And this is why I said we will find the optimal matching, because we will find the matching that minimize this expression. So this is the bottleneck distance. We have also another distance, which is the Wasserstein distance, that instead of taking the infimum here, we will say, instead of taking the maximum between the differences, we will take the sum of the differences. Also, when we define an optimal matching, so we will take all the differences x plus y plus 0 plus z. I'm sorry, and this is for a certain p. So here it is for p is equal to 1, but it can be like this is the pth Wasserstein distance. So yeah, basically those are the most common used distances to compare barcodes. So in my thesis, I applied this persistent homology pipeline to study proteins. So to give you an idea of what proteins are, so proteins are like macrobiological molecules that have different shapes, different sizes, and they are characterized by a 3D structure, and they have different functions in cells. They have different role in cells that goes like from transportating defense proteins, receptors, motors, etc. And the building blocks of proteins are what we call amino acids. So those are chemical compounds that have a general formula of, they have a central carbon that is linked to an amino group, a carboxylic group, and a side chain R that is specific to each amino acid. So this R side chain is different from an acid to another, and this side chain attributes to the amino acid a physicochemical property that could be like a positively charged amino acid, negatively charged amino acid, and other chemical properties, different chemical properties. So a protein could be an amino acid sequence that, due to the physicochemical properties of amino acids, it will fold over itself and attribute to the protein a 3D structure. But in biology, sequences have been very studied, and they are the more used than 3D structures. So biologists rely on a sequence to analyze it and understand the many biological process. One process that I am interested in is evolution. So during evolution, protein sequences undergo modification, and biologists compare protein sequences in order to retrace the evolutionary history of organisms. It means that if we have two sequences, they will compare the sequences and they will count the number of differences between these sequences. For example, here we have one difference, A that's become an S. It means that those sequences are similar, but there is only one difference. It means that those sequences are really close in evolution. And one of the questions that I was interested in my thesis is whether I can apply the same concept that is applied from biologists on sequences, but instead of studying sequences is to study 3D structures. Like biologists compare sequences, I want to compare structures in order to know if those structures are close or not. So basically, a protein structure could be represented by a point cloud, which is the atomic coordinates of the atoms forming this point cloud. And one limit of the sequence is that it is in a one-dimensional space. So when biologists want to study sequences, what they are looking at is only how the amino acid, how they will the this chain. They will not look at the interaction between amino acid. And to look at the interaction of amino acid, we have to consider the 3D structure. One limit was that we do not have enough structures to be able to compare them. For example, in 2024, today, there are approximately 250 million sequence comparing to 300,000 structures. But this is about to change because of the introduction of new algorithmic methods that are based on artificial intelligence, such as what we call AlphaFold, which predict a large amount of structures. And to be able to analyze this amount of structures, we have to apply methods that are easy to manipulate or process this huge number of data and be able to extract topological features and geometric features of this data. And this is why I was interested in applying topological data analysis on protein structures. In particular, I was interested in applying persistent homology on 3D structures of proteins. And to do this, so this is the overall pipeline that I applied. So from a structure, I consider the point cloud of the structure, apply the filtration process, and generate the geometric representation, which is the barcode that I just represented to you, to extract the geometric features of dimension 0, 1, and 2, so connected components, cycles, and cavities. And my question was, so I want to apply this to a collection of protein. From each protein sequence, I predict the structure using AlphaFold, and for each structure, I computed this geometric representation, which is the barcode. And my main question was to know that if persistent homology is reliable for computing the evolutionary distance between structures. Like I told you before, the evolutionary distance was to compute the difference between two sequences, the literal count the number of differences between two protein sequences. And I want to know if this distance is somehow correlated to the distance we compute from structures. In particular, when I say distance that we compute from structures, it means distances between the corresponding barcodes. So to do this, I applied this to a data set from 700 pairs of sequences. I computed the distances between sequences and computed the distance between the pair of barcodes using the Wasserstein distance. And I want to compare those two distances. And when I compared them, I obtained an estimation or a correlation of 0.78, which was not bad, which means that persistent homology could be a good start to use structures in order to estimate evolutionary distance, but it was not as good as other methods. Yeah, I will not go into the details. It is not as good as other methods that already exist in literature. So basically, there are indirect approaches that compare directly proteins. Indirectly proteins. There are direct approaches that compare directly proteins, and there are hybrid approaches that combine indirect and direct. And those three methods gave better estimation of the evolutionary distance. Um, so I wanted to do like go further in my methodology, and this required two improvement regarding the filtration process and regarding the geometric representation. So for the geometric representation, as I said, barcodes were considered to be a collection or collections of intervals. And I wanted to like really extract more information, not only intervals. I wanted to do some statistics on those barcodes. That's why I defined several measures on barcodes, such as the minimum birth value, the number of intervals, the mean of the length of intervals, the standard deviation, the skewness, the kurtosis, third and interquartile of d minus b, and the entropy in dimension 1 and in dimension 2. And this generated so using those statistics, I generated a 40-dimensional vector. Um, and I wanted to go beyond this. So I saw what is like generally done in literature, and I found that in 2015 there is Peter Bubenik who had like a similar intuition. He wanted to do some statistics on barcodes to be able to use them in like machine learning tasks. So he defined what is called persistent landscapes as also averages that are done on barcodes. And in literature, there exist also other type of curves that are called Betti curves. Like I said, Betti numbers, here Betti curves that count Betti numbers at each step of the filtration. And my idea was to combine, to see if I can combine the information coming from the statistics I defined on barcodes to combine the information provided by persistent landscapes and the information provided by Betti curves. And this is why I applied a discretization of persistent landscape into 40 value and a discretization of Betti curves into 40 values. And I combined the 40 values from the statistics, the 40 values of persistent landscapes, and the 40 values of Betti curves into a 120 dimensional vector that I called a biotopological marker. And this, my hope was that this constitute a like a more reliable representation of the protein structure, or I called it a vectorization of the protein structure. And really that intuition worked. So by combining all this information from the different geometric representation, I did the same process and I compared the BTMs that I call them BTMs. And this gave me a better correlation between evolutionary distance and this BTM distance, or the distance on BTM. And as I said, I wanted to go further into the method on filtration. So in all other work, I consider the point cloud as like just a point cloud, but only considering the coordinates of each point. And in order to have a better representation of the protein, I wanted to integrate biological information in the point cloud, such as the quality of prediction of the structure. And the second thing I want to consider was the density of the point cloud, because it also plays some sort of biological role, the density of this point cloud. And this is why I developed a weighting system that integrates those both factors, the prediction quality and the point cloud density. And the idea is that the higher the weight that I put on a point, the sooner it enters the filtration process. So here, for example, the points in red have the largest weight, so they enter sooner in the filtration, and then later on, other points begin to enter as the filtration goes, and those points have less weight. And by combining this with the BTM, we arrived to an estimation of 0.97 between the distance on BTM and the evolutionary distance. So, so I came to the conclusion that persistent homology first captures and can estimate the evolutionary distance from structure, and by developing a geometric representation that is more reliable and integrating biological information into this representation, we can refine the estimation of evolutionary distance. So this is part of the work I've done in my thesis. So I showed that persistent homology could be applied to biological data, and especially in proteins, and can be a good alternative of sequences to study the evolution of proteins. And this representation, so I will not present this here, but this vectorization, the BTM, I applied it with machine learning problems in order to predict other environmental conditions regarding proteins, like how the temperature affects proteins. And I studied also an extension of those representation in a spectral context, also that is related a little bit to persistent homology. I wish I can have another opportunity to present to you this work, but I guess this is what I want to represent to you today. Thank you for your attention.